lattice mode. Let's, yeah, zoom in a bit. A little bit more. Thank you. Yeah. I'm going to start by just bringing it closer and we'll see if the whole thing blows through or not. Let's come just a little bit wider, Al. Thank you.
It's across the front there.
Okay, then let's let go of it or stop slipping. Oh no.
Views?
Yeah, it might be hard to get that. You're right, it is just across the...
All right, we can put it in the forward box.
Okay.
Yeah, better than nothing.
Oh.
Maybe we'll see another circle piece somewhere else that'll be smaller.
You want to suck it, or do you want to?
Well, we finally got the right angle on trying to suck it, but now it seems...
I think it might get stuck.
Yeah, I think it's going to get stuck. Well, late now. Paul gets to clean the hose out.
Deal.
Turn it off and back on again.
We also come wide, Al. Thank you.
Yeah, stuck good.
Yeah, wrist is doing the thing where it spins.
Was it creeping?
Yeah. How do I?
You gotta go into calibrate. Set up, calibrate. 5, 5, 4, 2, 3. Wrist bias. You gotta, is it unfrozen?
Yeah, it's unfrozen.
Is it good? Not creepy?
Looks good.
Okay. Exit.
Cool.
Yep, stuck.
Should we try going to flush? Does that ruin the sample?
Silence of science.
Oh, we started the handover.
Okay, thank you. Well, it wasn't an exciting last dive, but it was still a pleasure to work with you all. Thanks so much for everything you've done for us over NA123 and all the work you'll continue to do into the future for everyone. This has been spectacular.
Thank you. It's been really fun working with you on shore. I think we've got a question now if it's okay to try to flush out this kelp into our flush jar.
I think we're...
Yes, absolutely.
We're doing it?
We're doing the flush jar.
Okay.
Yeah. Yeah. There'll be more kelp.
Still
totally
not
fine.
coming through. So.
I think the real story is I'll be cleaning the slurp hose in 4 hours or so.
So something to keep in mind for other samples on the next watch.
Yep. All right. I'll stow this then and then hop in the skin.
I was gonna say you could slurp some gravel, but I don't know.
Yeah.
Rocks are in short supply here.
I think because of the, I don't know what you call that part of the kelp where the stalk is. And
The bulb.
the bulb, yeah. The more stick-like part of it.
The pneumatocyst.
We still want a Niskin here also?
Yes, please.
Steve, are we still dropping or using a sample ID for that?
Yeah.
Back resist test? Okay. 151.
151 and 152. We're going Niskin 2
Roger.
for sample 152.
Got it?
Yep.
Cool.
Oh, the jaw creep is back.
It's just an annoyance. You're gonna have to calibrate it out again.
Calibrate, and then...
It's written right
Oh,
here.
got it.
5423.
this is the risk bias?
Yep. And then you have to, I think this, you might have to come out of it. I don't know. I don't know the secret ingredient there to making it set. You'd have to like re-index it, or you have to exit that. Just watch it.
It's still going a little bit.
Well, you'll never get it just right. It's just, it's too finely balanced.
Yeah.
It's kind of dumb. I don't know. It's always been like that.
All right. Well,
Never, you're not going to be able to get it so it's good all the time. You just kind of get used to having always fixed the wrist, you know?
yeah, yeah. All right, do we want to?
Well, we've got 4 minutes. Is there anything else here that we want to zoom on?
We can zoom on the basket star since we're doing a change.
Great.
Oh, yeah, I'm fully panned, so let me reposition a little bit.
Did you push the camera back out?
The current is not in our favor for these touchdowns. All right, let's zoom in.
On a shell or is that down there?
Yeah, it looks like it. I feel like they've been on like the rarely few like cobble that we've seen, like a random singular piece of rock that they've been on top of. They just find them and stay there.
Oh, they're really cool zoom. We bump a little to the left to get it in full, right?
Can we? It's a good question.
I know.
Yeah, I think that's a little bit to the left. All right, I think we're just about signing off here.
Sounds great. Thanks, everyone.
Yeah, thank you for
Thank you,
all the
guys.
dives.
Thanks for
Come back
executing them
anytime.
for us. Yes, please do. We'll have more for you. We'll go back to that
Hi,
sponge
guys.
reef. That was so cool. Hey, Di.
Hey, good
Hey,
morning.
Guy.
Good morning.
Do you
So
want
I
me to
noticed,
jump off so Lisa can
yeah,
join, or do you? Yeah. Okay.
either
I'll-
whichever you prefer.
Lizzie?
It's pretty darn simple. I know Lisa's listening, so it's pretty much just continue on to the next waypoint. The visibility isn't super great, but it's a lot better than it was. And honestly, we're not seeing a whole lot. The backscatter so far has been correct, flat, muddy bottom. So we've done
Okay.
2
And
transects.
what waypoint are you headed toward?
AZ.
AZ,
Which we
okay.
hope to be somewhat of a tiny rock feature. There is some harder return there on the backscatter. So we're ground truthing that part and then pretty much just get as far as you can before we have to pull, but do so safely.
Okay. Do you want us
It's
to?
fine with us over this habitat right now. Just go, you know, as fast as the vessel and the ROV team are comfortable going. I think we got up to 0.5 knots maybe on the last round. So if it stays as simple and they're comfortable or whatever they are comfortable with, just kind of zoom over to AZ. AZ and VA will probably be optimistically will be more interesting and have something to look at. So see how far you can make it.
And then I see the waypoints go from BB over to BC, due west. Is that still the plan?
If you can make it that far, you can go BA, BB, there's BB, and then BC. There's value in that because there's maybe something there, but also presumably you would go from a hard bottom to a soft bottom feature, and just covering that transition zone has value. So if you go that far, that's great, but don't worry about getting that far if not.
Okay. If you don't, no worries.
And I noticed no sediment cores. Is the bottom not conducive to that, or it's not a priority? Or I saw your scoop sample.
No, we'd definitely take some cores. I was kind of waiting to see some urchins. We had seen some earlier, and now, of course, we are not seeing any. So we did just sample some kelp. So we could take sediment cores here before you take off, just to get those in the bag. You can take 2.
Okay.
And they took a Niskin, so we would just need the sediment cores here just to make sure we have some, I guess.
Did you say to take 2 cores here? It was cutting out a little bit.
Yes, 2 would be good.
Well, maybe we will do that first before proceeding then, if that sounds good to you.
That might be best. Yeah, just because we collected the kelp, and might as well do the suite of
You want to switch over the cameras?
Here, just in case we don't encounter urchins, but we will. Taylor Ann will be on this shift, so I'm sure she'll tell you. She'll see what we have and haven't sampled, and hopefully we'll run into some more.
Okay.
Show me the weapon.
All right, I'm signing off, Lisa, if you want to jump on. Have a great last shift. Awesome working with you.
And open that box.
Opening.
Thanks so much.
Good.
Went really fast.
Yeah, we're shallow. Everything's going to be fast. They've taken no cores so far?
Correct.
No cores so far, but we'd like to take 2 sediment cores here before we proceed. Please.
Is there anything in there?
Good morning.
Float away?
I don't think so.
No, we haven't sampled much yet.
Roger.
Zoom out for us, Steve. Thanks.
Hey, good afternoon. It's Lisa here.
Hello.
Hey, Lisa.
Good afternoon.
Hey, Lisa.
Yes, afternoon.
Oh, it is afternoon.
Last afternoon. Yes.
I was
we've
still thinking
crossed
morning.
over we've crossed over the Rubicon.
Oh, this is neat to watch. I have not seen a sediment core done yet, so.
And we're doing the Bob Waters method, I believe, of sampling sediment cores, two at the same time.
It's about as far as it wants to go in. Oh, there it goes.
Use the muscle.
Yeah, got a little western with it there. get it to go that last bit. Lifted the vehicle up.
Wow. Lizzie, are you still with us?
I am. I figured I'm not in any hurry. Just soaking up the
cool.
last
Good.
little bit.
Good.
So who wants
Have
to
you-
take the explaining why we're doing these sediment cores?
I can.
I will defer.
Okay.
Uh-oh.
Oh, yeah.
Go for it.
So we just collected some kelp, I heard, which is of interest to me. I'm Taylor Ann, the data logger, and I'm doing some research on microplastics in the deep sea and in shallow waters. So my project idea started in kelp forests. It's seeing how kelp forests could potentially accumulate microplastics. And I want to see if kelp can act as a vector for these microplastics in the deep sea or other areas of the ocean. So we just collected a blade of kelp. I didn't see it. I don't know what it looked like. But yeah, so I'm going to analyze that piece of kelp to see if there's any microplastics stuck in its crevices or on its surface. And,
Zoom in just a bit, Steve. Get a snap. That's good.
Yep, got pictures.
Got it?
Yep.
Okay.
So I'll be analyzing that along with the push cores to see what microplastics are in the sediment, in the water column, and on that blade of kelp. And I'll also be looking at other organisms like sea pigs, sea pens, some urchins, and then I'll be doing this study again in kelp forests, not just in the deep sea. So the reason why I'm interested in kelp forests is because there's attenuated flow of water there due to the thick densities of kelp. So this basically just means that water flows slower in the kelp forests due to the presence of kelp and the water running into the kelp. So I hypothesize that potentially due to this slower flow and the water sitting in one place for a longer period of time, that this could allow for microplastics to accumulate, allow them to settle in the sediments, and then also potentially stick on the surfaces of kelp blades. And I'll be studying in Macrocystis perifera forests. So that's kind of the speedy, quick rundown.
What's happening with the? Are we just clearing out the? Okay. So Dan, do you want to explain what was happening there with the manipulator arm?
Yeah, core tube. So you can see the other plug there. The core didn't stay in the tube. So it's just trying to get the sediment out of it to try again.
Yeah, so rinsing it out.
Basically. Yeah.
So yeah, basically what's happening here is we're taking sediment cores, and the top of the core will be the most recently laid down sediment. And we can look at the different layers, and we can see what the environmental conditions were when those layers were deposited. So sometimes some of these sediment cores will be used for particulate organic matter analysis. We want to sort of know what kind of ecosystem dynamics.
It shoots up.
I think there's not any kind of plug at the bottom.
I don't know.
It's loose.
It could be
It
could be the valve on that particular core sampler.
Huh.
And so the idea with the core is to keep those layers in order intact in the core and not have it get mixed up. So if the sediment is too loose, it's not it gets mixed up, I guess, and those layers cannot be distinguished as well.
And as you can see, we already have that other core pushed in, and the reason we did that is because, as you can see, when we pulled up this core, it is spreading out sediments, and we don't want it to contaminate the top layer of the core that's already been pushed in.
Zoom in there for me, Steve. So we use a piece of rubber as a valve on the top, and it looks okay. Sometimes they don't seal in it. So it displaces the water when you push it in and should seal when you pull it out. It might just be the, okay, zoom out. Try one more time here.
And we are sharing these sediment cores with our partners at USGS. That's the United States Geological Survey. They're a partner with us on this cruise. We're sharing it with Taylor Ann. She was the one talking about her research project on microplastics. Taylor Ann, can you tell people what microplastics are?
Sure.
Seems to be a, yeah, great, thank you.
So microplastics are any small pieces of plastic under 5 millimeters in size. Some papers say 1 millimeter. And they can be either primary or secondary microplastics. So primary microplastics were intended to be created as small pieces of plastic. So when you think of microbeads from face washes or toothpaste, or even the fibers of our plastic-derived clothing that gets washed off, anytime you wash that type of clothing, it will end up in the ocean because there's no filters usually on our washers. Oh, it looks like it's too loose, huh? Sediment.
That one was almost stayed.
Yeah.
It might just be too loose. I don't know.
I'm
Maybe we should
gonna
find another area.
I'm gonna drop this on purpose and see what the other one does. And then if this one works, we'll pick up that other one and move to another location. Just some clear.
That sounds like a good plan.
And all through this, we have a little comb fish just beyond that basket star that's been staying put the whole time. That looks like it's getting buried a little bit.
Yes, he's taking notes. Doing a thesis on core sampling.
He'll be a pro by now.
Yes. And we always hold our breath, like, because it could just fall out, right?
Yeah.
Don't say that.
Okay. I'm not jinxing it. That's superstition. So Steve, the video person, just switched the cameras on the Herc to the view where they're going to now store the sediment core. This is on the starboard side or the port side?
Starboard.
Starboard, okay.
Oh, there's one.
There you go.
Great.
Thinking it might be something with that particular core tube.
Could try another one. I don't see us taking, we don't never take all the push cores, so.
Yeah, copy.
Yeah,
it's usually 2, so it's fine.
Yeah, we
why
haven't
do we
taken any yet?
No.
Not on this dive. Yeah. Why do we take 2?
So it's being split up between, I think, 3 different groups. So one group of interest in the microplastics, I believe, or in something, I'm not sure what it's for, is USGS is interested in the top 2, like maybe milliliters or so of the sediment. And then we have another lab, the Battelle lab, that's interested in microbes that could potentially be down here able to kind of digest or wither away microplastics. So that's something they're going to be studying. And then for me as well. So 3 different groups of interest for these cores.
Ow.
So that's why we take 2. So that everybody can get, because usually the top few centimeters is everything that we want because there's not so much mixing going on down here. Things pretty much stay uniform. So we are usually interested in those top few layers.
So that was push core 1?
Yes, push core number 1.
Okay, thank you.
And I just notate it as red tape. because I think our numbering scheme has changed from the past.
Oh,
so I think it was
thanks.
push core number 5, maybe. But
Oh,
yeah, so just notate the red tape and quiver position number 1. Yeah.
Thanks, Taylor.
Lifting the vehicle up again. That's full. That's both thrusters on.
So folks that are tuning in, we are on the north side of Santa Cruz Island, which is part of the Channel Islands here off of California. We're not too far from, we're offshore of Santa Barbara. And
I think
this
that
dive that we're doing,
I'm going to say that
is
that
mainly
tube
a
is
dive to
not
ground
sealing.
truth some of the mapping that had been done in this area. And I think, Rennie, are you here?
Yes.
Our navigator?
Yes.
Oh, shit. Can anybody hear me?
Hello?
Yeah.
We can hear you. I don't.
We can hear you.
Okay, sorry.
Yeah.
Sorry. So, Rennie, maybe you could explain what ground truthing of the mapping data is, since you're the guy who analyzes that data often, if you don't
Sure.
mind.
I think they're after taking a look at the backscatter and seeing how that compares. So the backscatter is the reflectivity of the turn, of the return, how loud the sound comes back, and if it comes back louder, it could be that it bounced off something harder, like a rock. We also know that it could be a layer of sediment over rock, but that's still the 30 kilohertz sonar still penetrates that layer of sediment and reflects off the rock. And if it comes back softer, it could be just a thick layer of mud. So oftentimes we want to take a look at if the backscatter is telling us what we think it's telling us by coming down here to ground truth it.
Yeah.
Yeah.
The maps
So that's
themselves
one
at
of
this
the-
depth are pretty accurate as far as like the actual
Still air in that thing.
Less about ground truthing that.
Yeah, so the sonar data that this is the sonars used to figure out what kind of substrate type there is, and that's what Rennie was just explaining. Is it hard bottom? Is it soft? Or is it maybe mixed? And the sonar data also lets us know how deep the seafloor is from the surface, so we can get bathymetry and depths. And we are often looking for hard bottom, because hard bottom can maintain the growth of filter feeders like deep sea corals and sponges, and they in turn, as they grow, form more complex habitat with the fans or the sponges that they are growing, and that becomes habitat for other animals, invertebrates and fishes. And they're beautiful. But this soft bottom habitat is also important. I think it might seem less diverse and exciting, but if we were to look at the sediment cores, not only for the geology, the type of rock that's there, we would find a lot of what we call infaunal organisms, and these are animals that live and burrow in this soft sediment. And we can see some of them, like the sea pen that you can see, the white stick that's sticking up out of the substrate here.
Which is a coral.
Yes.
Yeah. Those sea pens are corals that specialize in, they have a specialized peduncle, I think it's called, that anchors them in soft sediment. And most of the other corals need hard substrata to attach to.
Oh, great zoom in, Steve, to sort of show that there's all kinds of small holes. These are openings for the different organisms that are living inside of the soft sediment here.
Okay, close her up.
Many of them are worms, different kinds of worms. Just like on land, earthworms.
Yeah. So off the west coast, only about 10% of the seafloor out to, from the shelf, continental shelf area, out to 3,000 meters is only about 10% of that area is estimated to be hard and mixed substrata. So most of the seafloor is soft substrata like this.
South?
Yep. Are we ready to move on from here?
Yeah, our next waypoint is AZ.
That's right.
I have a quick question for Teeleran. Which tape color is for push core 2?
The second one was orange tape, and it has number 3 on it.
Thank you.
I'm going to center up the Beeble.
Bridge nav.
You can change Argus heading to south.
100 meters, bearing 172. Thank you.
And those push cores were sample number 153 and 154.
Thank you, Teeleran. Got it.
We have the speed set to 0.5 right now.
Yep.
Okay. That's
fine.
Good.
Come down 5 meters on Argus.
Coming down.
That's a little half-banded rockfish. Many of the rockfish.
I was wondering if I saw those. Cool.
Yeah. The little. Go ahead. Most of the rockfish, well, the common name, they're referred to as rockfish because many of them prefer rock and complex rock habitat. But there are some that occur over mixed substrata and even soft. We're kind of getting into more of a mixed area right now. These are are these shells or cobbles?
That's what I was wondering, if they were clam shells.
Zoom in, Steve.
They look like it. Anyway, half-banded rockfish are one of those that are, and that's what these are out in front of us with that sort of, they kind of have a diamond dark spot on their sides. And they're one of the dwarf rockfish species. They don't get very big.
These do look like shells.
Yeah.
And the shells themselves become hard habitat for a
providing
filter feeder to land on. And you can see there's a hydroid, I think, on one of the shells that we just passed.
Yeah.
A bunch of these rockfish.
Hydroid. What's a
Daniel's
hydroid?
saying they're most likely.
Yeah, it's a hydroid.
Cesta.
What did you say, Lizzie? I'm sorry.
Oh, Daniel in the science chat was saying these are likely Cesta shells.
Oh, okay.
So
that we were seeing overnight in deeper water, but over in the 500-meter range.
Yes.
Okay.
Okay. Thank you. I think Dan, our pilot, asked what a hydroid is.
Yeah.
Gosh, it's a it's an invert. I know that.
I'm more of a fish person, so I can't answer that one very well.
Are they in the same, they're not an octocoral, but they are like an anemone? I'm going to look it up.
Stand by, Dan.
Yeah, the power of Google.
So, from what I remember, they're I know they're hydrozoa and they're like predators related to jellyfish.
Yes, so think of jellyfish as flipped over. So the jellyfish are the ones that live in the water column and they have the tentacles and they have that sort of gelatinous body. Well, the hydroids are if the jelly had flipped over and landed and formed a stalk. And so it's still very gelatinous and it has those tentacles that it uses to sting its prey and feed on.
I see.
So they have a polyp, just like the deep sea corals that we were looking at, but they don't have the calcareous skeleton. They're very jelly-like.
Little comb fish we just went over. So that's what we're seeing fish-wise. There's some comb fishes and then these half-banded rockfish. And these sea pens, I do not know the species. Anybody?
I'm trying to look that up now.
Quick zoom there.
Okay, thanks.
Zoom and zip.
Sea pens or sea
Zoom
whips.
and zip.
They get referred to as they get larger like this and taller.
And you can see in the polyps here on the.
And as their density increases in these soft sediment habitats, they provide structure. And there's some thought that they, in denser congregations, they may provide nursery habitat for certain fishes, their eggs and larvae. There's sort of an island, anywhere there's structure, you see things gathering. You see more things gathering. Like when we were on soft sediment on some of these dives for long periods of time and you come upon a single rock and it's covered with all sorts of things that have settled on it and a fish or two hiding.
There's the basket star, just like when we saw it during the watch change.
Yep.
So our heading is south, correct? We're going south.
Correct.
Somewhat.
Yeah.
Yeah. We're heading towards the island.
Yeah.
Correct.
And we're at 90 meters depth. And the temperature is 11 degrees centigrade on the
really?
Nautilus Live. Yeah, which is
water temperature?
Kind of warm.
Yeah,
that's what I'm seeing.
Not that cold at all.
No, 11 degrees C. On the Nautilus Live website, of course, where the public is watching this video from, their temperature is displayed in both degrees Fahrenheit and centigrade, as is, and then the depth is displayed it switches from meters to feet. More comb fish we're going by, and there's a little flatfish. It's hard to see, but sand dab comes to mind.
Which are good to eat.
Yep.
I said I was going to move those lights up last night. I didn't.
These two green dots in the center of the screen are lasers, and they're spaced 10 centimeters apart. They help us. 10 centimeters is about 4 inches, and they help us estimate the size of things. So I have a question for navigation.
Yep.
Is how far are we from the last transect?
We are 200 meters from the last transect.
Oh, we are. Okay. So we could start another transect, and it's Rennie. Thank you.
Yeah, we can
start
that.
Another transect as we go along.
We're going 172.
172, roger.
Okay with that, Lisa?
Yeah, that's great. And maybe once we start the transect, we can do a round of introduction since we just did the watch change and there's a new slate of folks on watch.
When is this 50?
Add 200 meters, 172. Thank you.
More work.
Begin transect.
50 meter steps, more work.
What's that?
More work at 50 meter steps.
And I guess I like to push buttons.
Which number is this? 3?
Yes, transect number 3.
Roger that.
Hey Lisa, this is Lizzie. Do we still have a briefing call today? I know we don't have any more dives, but I wonder if Nicole would still want to chat about anything.
Oh, that's right. Thank you for reminding me.
Well, I didn't even know if if it's even a thing still.
It's not scheduled.
I have something on my calendar, but I mean, I don't know if it's,
Well, why don't you and I both just call in and see what's going on and then pop back in if we need to.
Okay, that sounds good.
So, yeah, Nicole just chimed in on the chat to say hi.
Oh, hi.
Nicole. Yeah.
Oh, we don't need to have a call.
She said we don't need. Yeah.
Okay, but we will not leave. We will stay.
Can you press
Unless
the
we
transect button, please?
Transect button.
I think I'll just respond to her in the chat.
Thank you. Just gonna slow down a bit here for a minute to get Argus a little closer so we can have a better visual on ROV. ROV Hercules. It's incredibly flat here.
Yeah, definitely.
Our depth hasn't changed.
And now we're seeing urchins now that we're on transect.
Yes, is that always the case?
Mm.
Very large sea star down
there. So maybe we could do a round of a round of introductions. I'll start. My name is Lisa Vonink, and we are just passing an octopus there. Very cute. I work for the Office of National Marine Sanctuaries in the West Coast Regional Office, representing the 5 West Coast sanctuaries. Nautilus has done several cruises in our national marine sanctuaries, Olympic Coast, Monterey Bay, and right now we're in Channel Islands, but also in proposed national marine sanctuaries like the Chumash Heritage National Marine Sanctuary. And I'm the policy coordinator, and it's been great for me to get back to my science roots and be part of this deep sea exploration.
And I'm Diana Waters. I'm with the National Marine Fisheries Service lab in Santa Cruz, California, and our group studies seafloor habitats and ground fishes. And this is my first involvement with the Nautilus cruise, and it's been fantastic. My group does visual surveys has used other vehicles in the past, but this is our first time working in this way with the Nautilus. So.
Lizzie Duncan is also here. I'm a leftover from the previous watch. I'm just joining in for a few minutes since we're almost finished with NA123 in the Channel Islands. which I work for the Channel Islands National Marine Sanctuary, so I'm just observing, and I'll probably have to go soon because it's lunchtime, but it's been so nice to have Di and Lisa leading watches for this portion of the cruise. And so I just want to say I really appreciate them and all the Nautilus crew for everything they've done.
We appreciate you, Lizzie. Thanks for putting together a great cruise in Channel Islands. Really fantastic.
Thanks, Lizzie.
Yeah. Great job, Lizzie.
Shucks. Thanks, guys.
And now the hard work begins because we've collected massive amounts of data, which our partners are going to help us analyze, and then we're going to write characterization reports trying to describe what we found, and all this information will be used to help us make more informed management decisions about how to manage uses in the sanctuary and how to better protect some of the more fragile habitats. So this is just the beginning.
Yeah. And many of these areas we've been diving on during this cruise have been located. We've been able to locate them. Lizzie has, and plan her dives based on this new mapping effort that's been going on. And so you need good maps to locate where you want to dive and focus your efforts. And the high resolution maps have not been available until recent years with all the effort that's been going on, thanks to Chris Caldo and others that have created this, especially in the Southern California Bight, a mapping initiative.
Right, and the Nautilus ship and their crew have participated in a lot of that mapping, and maybe now's the time for us to hear from the folks who really make this happen. We would not be able to broadcast this imagery to you without the crew that are making this happen.
Here in the control van, this is Stephen Matter in the video chair. And this is my last watch of the season. So I want to thank Lisa and Di and all my shipmates here in the van. It's been a good run.
It definitely has been, Steve. It's been awesome.
Been great working with you. Yep, awesome.
This is Jake over here, flying Argus.
Dan in the Herc chair.
This is Rennie in the navigator chair.
And this is Taylor in the data logger seat, and this is also my last watch. So I'm really thankful to have been able to come out this season as a science manager in training and also collect some samples for my master's thesis project. It's been a great experience, and I love being out on the Nautilus. So enjoying my last watch.
Thanks, Taylor Ann. And we look forward to working with you in the future. You're not far from a lot of our national marine sanctuaries, so we look forward to having you come and give a talk to one of our advisory councils about what you found.
Sure.
Because we're very interested in a better understanding the impacts of microplastics on the marine environment, particularly in sanctuaries, and you collected a lot of your data inside sanctuaries. So
yes, yeah, I would be more than happy to. Yeah, of course,
so
give back.
Let's stay in touch.
Yes. Okay, sounds good.
Yeah. Taylor, maybe you can explain to folks who are interested in sort of getting involved in this type of exploration, like how did you start working or engaging with the Ocean Exploration Trust? Because this is your second year, if I understand.
Yeah.
Maybe you could tell people what your pathway was to being part of this.
Yeah. I'd say I definitely had not a roller coaster of a pathway, but a windy pathway into ocean science. I grew up in the Midwest, so I didn't get the chance to study ocean science or marine science at my university in Indiana like I had wanted to, so I just got a general biology degree. Oh, here come a lot of urchins. Is that what this is?
Totally.
Yes.
Urchin City.
Is it? Really?
Yeah, looks like it.
Wow.
It looks like it to me too.
Yeah.
Yep.
So, sorry, I got distracted.
Ooh, urchins.
So yeah, I got my general biology degree in Indiana, and there I did some research in a little lab that focused on mostly, I guess I just counted bacteria my whole time there. I didn't really gain the skills that I really wanted to, but it gave me the insight to see what it was like to work in a lab, and I knew I liked that, so that was at least good. And then I moved on from Ball State after 4 years there, and I finally got a scholarship in something marine because it was really hard to get a scholarship or an internship in marine science with no experience in marine science, but an interest and a passion for it. But I got a scholarship to attend Duke University Marine Lab for a semester where I was able to focus on my own independent research on ocean acidification in collaboration with Dr. Zachary Johnson's lab. And then I also took some courses because this was after I graduated undergrad. So I got some graduate level courses in marine sciences. So marine policy, marine invertebrate zoology, mammalogy. It was a really great time. And I got to go to Panama and do a travel course there with Duke. And then we also did a little independent research project there. So I finally was getting into my little niche. And that took about 4 years of hard work in undergrad and having that interest, and finally had the little pathway to open up. I applied for many other things and got told no, either because I didn't have experience yet, which was like, how do you get experience if no one gives you a chance?
That's right. It's
yeah,
a catch-22 sometimes.
It really was. But I knew that my passion and my willingness to do the work that I wanted to do was something that was, like, set me apart, I guess, from other people and would be useful. So I just kept trying. And then, yeah, eventually it happened. And then when I was at Duke, I had this awesome professor that let me nerd out in class and after class and bring things into the lab when I just go exploring and find little marine invertebrates and look at them under the microscope. And her name was Dr. Cindy Vandover. And she was really nice to me and answered all my questions about everything. And then I found out that she was a deep sea researcher. And I started asking her tons of questions about the deep sea. And she was like, you know, the best way for you to answer all these questions is to just, you know, get into deep sea research yourself or, you know, get some experience out there. And I was like, oh, yeah, that would be cool. I wish that was possible. And she's like, oh, it is. I know about these different organizations. And here, I'll give you, you know, their websites. You should apply. And so she told me about the Nautilus and Ocean Exploration Trust, and I applied for the ocean science internship. I didn't think that I would get something that cool, but I, you know, applied for it anyway. And a few months or weeks later, I found out that I got asked for an interview, and the interview went really well. The team was great, and I'm typically a really shy person, which I know people get surprised by that, but I didn't feel shy during that interview, so it was great. And then, yeah, I got accepted as an ocean science intern and came out for about 18 days last year, I believe, and had the similar role to what I have now. And then I got asked to come back again this year after, you know, kind of poking Nicole to be like, hey, I really enjoyed my time. Can I help you again? And yeah, turned out that I was able to come out again. So yeah, just being persistent and passionate. That's kind of how I got out here.
Yes, I think your pathway, your perseverance is a great example of what can happen if you just follow your passion.
Mm-hmm. Yeah.
And when you get back to Cal State University Northridge, please say hi to Larry Allen.
Of course, yeah. That's yeah. So it's such a small world. I'll definitely tell him that you say hello.
Yes, him and Michael Franklin.
Awesome.
Spent a lot of time with him on the research vessel Yellowfin.
Oh, that's awesome. Yeah, I've been on the Yellow